Hello, everyone. My name is William, and I'm going to be going over my presentation on the regulation of glycolysis and gluconeogenesis. This is for CHM 530, Concepts of Biochemistry. In this presentation, I am going to discuss what glycolysis and glucogenesis are and their pathways, how aldolase affects each of these pathways, the different isoforms of aldolase, and their different expression patterns. And finally, I'm going to discuss how aldolase B expression can be regulated by transcription factors such as HNF1, HNF3, CEPB, and BP. So to start this presentation, I'm going to begin by talking about glucose. Glucose is the basic molecule that all of the principles are based upon. Glucose is the most common molecule that is used for cellular energy. Glucose is broken down into energy in a process that is known as cellular respiration. This process begins with glycolysis, where glucose is first broken down. Its products then enter the citric acid cycle, which products then enter the electron transport chain. This presentation is going to focus on the first phase of cellular respiration, which is glycolysis. While glycolysis is an essential stage of cellular respiration, the majority of energy that is produced during the electron transport chain phase of cellular respiration. So glycolysis. Glycolysis is the first stage of cellular respiration. This is when glucose is utilized as an energy source. Essentially, one glucose is broken down by utilizing 2 ATP, and it results in the production of 4 ATP, 2 NADH, and 2 pyruvate molecules. And that's for each glucose molecule that goes through glycolysis. The pyruvate can then be used in the citric acid cycle. The majority of energy that is produced during cellular respiration is produced during during the following phase, which is the citric acid cycle. During glycolysis, a total of 4 ATP are produced. However, it takes 2 ATP to convert glucose into ATP, so there's only a net positive of 2 ATP. In contrast, during the citric acid cycle, there is a net positive of 30 to 32 ATP. However, this is an aerobic process that requires the use of oxygen, where glycolysis can occur with or without the presence of oxygen. The glycolysis process generally occurs in the cytosol of the cell. The glycolysis pathway. The glycolysis pathway is separated into two main phases. In the initial phase, 2 ATP are utilized, but in the secondary phase, 4 ATP are produced, along with 2 pyruvate molecules and also 2 NADH molecules. This equates in a net positive of 2 ATP, which is not substantial when you compare it to the total ATP output of the entire cellular respiration cycle, but the 2 pyruvate and the 2 NADH molecules that are created are essential for the process to continue. This process begins by glucose being converted into glucose 6-phosphate by an enzyme known as hexokinase, and the use of 1 ATP. The ATP is converted into ATP during this step. Glucose 6-phosphate is then converted into fructose 6-phosphate by an enzyme called phosphoglucose isomerase. Next, in the third reaction, this is where fructose 6-phosphate is converted into fructose 1,6-bisphosphate by an enzyme that is known as phosphofructokinase. This step requires the second and final ATP of the entire process. Next, aldolase converts fructose 1,6-bisphosphate into two separate 3-carbon molecules. These two molecules are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. As the image shows to the side, the dihydroxyacetone phosphate molecule cannot continue in the glycolysis pathway the dihydroxyacetone phosphate will be converted into a glyceraldehyde 3-phosphate by an enzyme called triphosphate isomerase. Because the dihydroxyacetone phosphate is being converted into the glyceraldehyde 3-phosphate, there will now be a total of 2 glyceraldehyde 3-phosphates that will go through the rest of glycolysis from just the one original glucose molecule. The glyceride 3-phosphate can then be converted into 1,3-bisphosphate glycerate by glyceraldehyde 3-phosphate dehydrogenase. During this phase, NAD is converted into NADH, which is an essential molecule for the electron transport chain. To clarify, 1ADH is produced for each molecule of the glyceraldehyde 3-phosphate. Next, the 1,3-bisphosphoglycerate is converted into the 3-phosphoglycerate by another enzyme called phosphoglycerate kinase. And during this process, one ATP is created. ADP is converted into ATP during this phase. Then the 3-phosphoglycerate is converted by an enzyme called phosphoglycerate mutase into 2-phosphoglycerate. This is then converted into phosphophenyl pyruvate by an enzyme called enolase. And finally, in the last step, the 10th step of the process, the phosphophenyl pyruvate is converted into pyruvate by an enzyme called pyruvate kinase. During the final stage, another ADP is converted into ATP. And again, just to clarify, the one ATP is produced for each of the glyceraldehyde 3-phosphates that went through glycolysis. So, meaning for one glucose that entered glycolysis, there would be 2 ATPs produced in the stage. A total of 4 for the entire process. The glycogenesis pathway. Glycogenesis is essentially the reverse process of glycolysis. However, there are a few steps that are different. When prolonged fasting or vigorous physical activity occur, glycogen stores can be depleted. The body then has to find another source of fuel. In the process of gluconeogenesis, the body is able to create glucose from other molecules. These other molecules can be like glycerol, lactate, pyruvate, or even amino acids. Because there are certain enzymes required for this process, the area where it can occur are limited to just to the liver and the renal cortexes. And the exact process of gluconeogenesis is largely dependent on what the molecule is that is being converted into glucose. For example, the initial enzyme will differ depending on what you are starting with. The enzyme that's going to convert pyruvate into glucose is going to be a different starting enzyme than one that would transform like lactate into glucose. So the enzymes that are required in the initial steps depend on what molecules being converted into glucose. The majority of this process is the same as glycolysis. There's just 3 different steps. But since this presentation is focusing on aldolase, I'm going to focus on that. In this step, there's a reversible reaction of what we talked about earlier. On the next slide, I'm going to get into aldolase. Aldolase is an essential enzyme in the process of both glycolysis and glucogenesis. It is the enzyme that is responsible for the reversible conversion of fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate during glycolysis. In glucogenesis, dihydroxyacetone phosphate is converted into fructose 1,6 bisphosphate. These reactions are inverses, except that fructose 1,6 biphosphate can be converted into two different molecules. But the fructose 1,6 bisphosphate can only be created from the dihydroxyacetone phosphate. So, in other words, during glucogenesis, the glyceraldehyde 3 phosphate needs to first be converted into the dihydroxyacetone phosphate before aldolase is able to convert it back into the fructose 1,6 bisphosphate. So while the fructose 1,6 bisphosphate can turn into two molecules, only one of the two molecules it produces can turn back into the fructose 1,6 bisphosphate. So isoforms of aldolase. There are two overall classes of aldolase. Um, However, there are multiple subgroups. The two main classes are divided into class 1 and into class 2. Class 1 aldolases are found in all living organisms. This is from prokaryotes and eukaryotes all the way up to plants and animals. In, in contrast, class 2 aldolases are only found in certain species like fungi and bacteria. The class 2 aldolases are not found in higher level organisms like animals and plants. Class 2 aldehydes also require a metal cofactor in them. Class 1, we're going to focus on class 1 aldolases for this presentation since we are talking about humans in this case. So class 1 aldolases can further be broken down into the three subgroups that have been found in humans. We are going to focus on the class 1 aldolases since they pertain to humans. So each of the different forms of aldolases are found in different areas of the body. The three different types are type A, type B, and type C. And each one of the different types is found in a different area and they all serve a different function. So type A is found throughout the skeletal muscle. Type B is found in the liver and kidneys. And finally, type C is found in the brain and also it is in other neurological tissues. These enzymes also have different functions. For example, type B is able to break down fructose as well as participate in glycolysis and glucogenesis. The other two types, type A and B, A and C, are unable to process fructose. So aldolase B expression. So as I previously mentioned, aldolase B is part of the family of class 1 aldolases. This is the class that is found in mammals. And then specifically, the class B type of aldolase is found in the liver and then also in the kidneys. The class B aldolase is able to process fructose on top of its normal functions. It's one of the things that separates it from the other two. Aldolase B is also utilized in a number of different processes. These processes include glycolysis and gluconeogenesis, as we discussed earlier. The upregulation of glycolysis, or I guess, in order to maintain adequate blood sugar, the process of glycolysis and glucogenesis are reversely related. So they both can't be occurring at the same time because they serve their opposite they have opposite purposes. So if glycolysis was happening and glucogenesis was happening at the same time, it would be getting nowhere because the glucose would be getting broken down, but then other things would be getting turned into glucose. So there's different factors that help upregulate either glycolysis or glucogenesis. So one example of that is insulin. So insulin causes the upregulation of glycolysis. And this is because insulin helps bring glucose into cells to be broken down into ATP and used for energy. So as the glucose in the blood is being utilized, the blood glucose level is going to drop, which can lead to hypoglycemia. So the insulin stimulates the process of glycolysis to happen so that the patient doesn't become hypoglycemic. Um, and then that's going to be the upregulation of glycolysis. And because there's an upregulation of glycolysis, there would be a downregulation of gluconeogenesis. Um, and then inversely, there would be a deregulation of glycolysis and an upregulation of gluconeogenesis when someone is at a point where they're fasting. So maybe they went to sleep for the night, um, and there's no longer a high demand for energy, then the glycolysis process needs to downregulate because there is not a high demand for the glucose. So in order to help do this, there are enzyme or messengers in the body, like glucagon, that when they're released, they help signal that the glycolysis process needs to downregulate in order to make sure that the blood sugar level doesn't drop too quickly. And then here are the references I used for my presentation.